This is lecture 27 on fatty acid and amino acid catabolism. In the last lecture, we covered carbohydrate catabolism, and we showed how one glucose molecule is catabolized and broken down both through aerobic and anaerobic processes. This included glycolysis, which occurred in the cytoplasm in the absence of oxygen, and also through the citric acid cycle and the electron transport chain that included oxidative phosphorylation, which required oxygen and was under aerobic processes. Through these reactions, one molecule of glucose produced 38 ATP. Now we're going to look at fatty acid and amino acid catabolism, and we're going to see how intricately interwoven they are with carbohydrate metabolism. And one of the most surprising things that many students have is how the interrelationships between carbohydrates, fatty acids, and amino acids are in our metabolism. So let's begin. So we're going to look and compare different sources of lipids and describe the catabolism and also the anabolism of lipids through a special process called beta oxidation. So when you think of beta oxidation, you should think of the catabolism and anabolism of lipids. We'll look at the dietary sources of proteins and then also look at the different examples of protein catabolism and anabolism. And they use a special process called deamination. Okay, let's begin at the basics on fatty acid catabolism. Most fats exist as triglycerides. You learned this in SCB 203. So this is new. Now, enzymes called lipases help break down fatty acids through lipolysis into fatty acids and glycerol. Okay, remember the glycerol is the 3-carbon backbone that attaches to 3 fatty acids to form triglycerides. Now, the catabolism of glycerol results into its conversion of glyceraldehyde 3-phosphate. And we know this already because this is an intermediate in glycolysis. So immediately we see that fatty acid catabolism, it enters the catabolic reactions of carbohydrates. So if you remember this figure of glycolysis, I've put a star right here next to glyceraldehyde 3-phosphate. And this is where glycerol will enter glycolysis and then also result in the formation of ATP. Now, fatty acid catabolism is unique because most of the energy from triglycerides comes through a process called beta oxidation. Okay, as a result of beta oxidation, we produce a molecule called acetyl-CoA, which we saw already entering the Krebs cycle or the citric acid cycle. And as a process, ketone bodies are produced through ketogenesis. And we mentioned ketone bodies briefly in previous lectures, and we'll talk about it more this time. Let's explain what beta oxidation is. Beta oxidation are the reactions in which fatty acids are oxidized in the mitochondrial matrix of the heart and skeletal muscle. The heart and skeletal muscle are specialized to be able to use fatty acids, and they do this through a process called beta oxidation. Now, what happens during beta oxidation? Well, in beta oxidation, fatty acids are bound to coenzyme A. Okay, we've heard about coenzyme A before, and the oxidation goes through this process called beta oxidation. Okay, so here we have a fatty acid here. Okay, right here, and it interacts with coenzyme A. So this is the first reaction. So we end up with the fatty acid combined with coenzyme A. The next step is the fatty acid chain is oxidized by NAD and FAD, and 2 carbons are removed. Okay, so beta oxidation, 2 carbons are removed. So here we have the oxidation by NAD and FAD. Okay, and 2 carbons are removed. Okay, and the 2 carbons are removed form acetyl-CoA. Okay, so FADH2, NADH, and acetyl-CoA and a fatty acid with 2 fewer carbons are produced. Remember, the 2 carbons are removed to form acetyl-CoA, and so now our fatty acids are 2 carbons shorter. And FADH2 and ADH goes to the electron transport chain to form ATP. And acetyl-CoA, as you might expect, goes to the citric acid cycle. And the remaining fatty acid is further oxidized. So here in this diagram, we see the products of beta oxidation are FADH2, NADH, acetyl-CoA, and the fatty acids. FADH2 and NADH go to the electron transport chain, and the acetyl-CoA goes to the citric acid cycle. Now what happens? Well, another CoA is added to the shorter fatty acid. And so beta oxidation is repeated until completely converted to acetyl-CoA. And then each acetyl-CoA enters the citric acid cycle. And as before, NADH and FADH2 enter the electron transport chain and ATP are produced. Okay? So here we see the acetyl-CoA binds to the shorter fatty acid, and beta oxidation continues until the fatty acid has been converted to acetyl-CoA subunits. So beta oxidation will occur until there are 2 carbons left. And that final 2 carbons forms acetyl-CoA and enters the citric acid cycle. So there will always be one round less of beta oxidation until there's 2 carbons left. Okay, so for example, if we have palmitic acid, which is composed of 16 carbons, beta oxidation will occur 7 times, because after the 7th time, you're down to 2 carbons left, and that doesn't need to be oxidized anymore. That will just bind to us. That will be a form of acetyl-CoA itself. Okay. As a result of the beta oxidation of palmitic acid, will produce 7 NADH molecules and 7 FADH2 molecules for each of the 7 beta oxidation reactions. And this will produce 35 ATPs. Remember that 3 ATPs are produced by NADH and 2 ATPs are produced for each FADH2. So 7 times 3 is 21. 7 times 2 is 14. 14 plus 21 equals 35 ATP. So that's how we can account for these electron carriers and the formation of ATP in the electron transport chain. Now, 8 acetyl-CoA molecules are produced. Remember, 7 through beta oxidation, and then there's 1 2-carbon molecule left that forms acetyl-CoA. So there'll be a total of 8 acetyl-CoA molecules that enter the citric acid cycle to generate 96 more ATP. Now remember, in the citric acid cycle, there are 3 NADH produced, 1 FADH2 produced, and 1 ATP produced. So for each of the 3 NADHs, this occurs 8 times 24, and we get 3 ATPs produced. So 24 times 3 equals 72 ATPs. 1 FAD is produced in the citric acid cycle, but this occurs 8 times in the beta oxidation of palmitic acid, and for each of these we get 2 ATPs. So 8 times 2 equals 16. And finally we have 8 ATP produced by state-dependent phosphorylation. And of course that results in 8 ATP. So 72 ATP plus 16 ATP plus 8 ATP produces a total of 96 ATPs from the oxidation of palmitic acid. From this, we see that there's much more ATP produced from a fatty acid than there are from glycolysis of glucose. Now in the process, we said ketogenesis occurs. and it's a process in the liver producing ketone bodies from acetyl-CoA. So there is a effect of going through beta oxidation, and that's the production of ketone bodies. And this occurs in the mitochondrial matrix of liver cells. Two acetyl-CoA molecules combine to form a 4-carbon ketone body. And the liver cannot metabolize ketone bodies and releases it into the blood. But heart and skeletal muscle can produce metabolism in the heart produce two acetyl-CoA. And the acetyl-CoA enters the citric acid cycle and oxidized. The brain cannot use ketone bodies. It usually relies exclusively on glucose, except under starvation conditions. Now, the liver produces large amounts during extreme caloric restrictions or starvation. And these ketone bodies may accumulate in blood, producing what is called ketosis. And then the ketosis may decrease the blood pH, producing ketoacidosis, and this could be, dangerously low acidification. What happens during amino acid catabolism? Well, proteins that are used for catabolism are generally dietary or already present in the cytosol. Okay, and of course, it's important that blood protein levels are within the normal limits. Okay, the amino acids enter hepatocytes, and the amino group is ruled by transamination. Okay, so transamination is the removal of the amino group of amino acid. Remember, an amino acid has two functional groups: a carboxylic acid on one end and an amino group at the other end. Transamination is the removal of the amino group, and this is transferred to a 5-carbon alpha ketoglutarate. Okay, and alpha ketoglutarate can be converted by transamination into glutamate. Okay, and so you end up with two products: a carbon skeleton and the amino acid glutamate. Okay, and so here's our glutamate here and the carbon skeleton here. Okay, and of course, the carbon skeleton may be converted to pyruvate, acetyl-CoA, glucose, or other citric acid compounds. So the fate of the carbon skeleton may have many different possibilities, and they are oxidized by glycolysis or citric acid cycle. Again, we're seeing the intermixing of the reactions between the carbohydrates, fatty acids, and now proteins. Okay, so here the carbon skeleton can be it can be modified into a variety of compounds, which then can be oxidized. Glutamate, on the other hand, undergoes oxidative deamination in hepatocyte mitochondria, and the removal of the amino group forms ammonia, okay, which is toxic, and reforms alpha ketoglutarate. Now, alpha ketoglutarate we know because enter the undergo oxidative deamination in the mitochondria. Okay, and some of the amino groups are produced to make new amino acids, or the ammonia can be produced from the removal of the amino group to form new amino acids, or formation of urea. Okay, so some amino groups are removed as ammonia to form new amino acids, and others, the ammonia is toxic, and to remove the toxicity, it enters what's called the urea cycle. When urea is much more not as toxic, and two ammonia groups combine with carbon dioxide to form urea, and most of this is eliminated in the urine. Okay, so this is the process of the oxidation of an amino acid. Again, there's intermixing between the reactions of fatty acids and carbohydrates, but there's also unique reactions of amino acids, namely transamination, as well as deamination and the formation of urea. So let's look at the big picture of nutrient catabolism now that we've looked at all three different major organic molecules. So we have lipids, carbohydrates, and proteins. Okay, so carbohydrates we started with the oxidation of glucose to form glyceraldehyde 3-phosphate, into pyruvate from glycolysis, into acetyl-CoA as it enters the citric acid cycle, and then to the electron transport chain and oxidative phosphorylation. Okay, and so we saw that 38 ATPs are produced. Fatty acid oxidation, we saw that this occurs through beta oxidation, which produces a number of NADH and NADH2 that enters the electron transport chain, as well as intermediates of acetyl-CoA that enters the citric acid cycle. Glycerol, on the other hand, is converted to glyceraldehyde 3-phosphate that enters glycolysis. Finally, proteins are digested to amino acids, and through transamination, they have a number of possibilities of entering the glycolysis, entering the intermediate reactions before the citric acid cycle, or entering the citric acid cycle. So what I like about this figure is that this shows how the interreactions of the three major organic molecules are when they're oxidized. And each of these can be interconverted to one another, which is really pretty fascinating. Okay, let's look at fatty acid anabolism now, the buildup of fatty acids. Okay, well, it's pretty much beta oxidation in reverse. Okay, so fatty acid anabolism, another name for it is called lipogenesis. Okay, and it uses an enzyme called fatty acid synthase, and this occurs in the cytosol. And just like beta oxidation reduced fatty acids by 2 carbon molecules, fatty acid synthase lengthens fatty acid chains by 2 carbon units. And the fatty acids are attached to glycerol in the endoplasmic reticulum and stored as triglycerides in adipocytes. Okay. The fatty acids often start out as other molecules, such as glucose or amino acids, because excess glucose will be converted into glycerol and amino acids into triglycerides. So there is even interconversion during the anabolic reactions between proteins, carbohydrates, and fatty acids. What about amino acid anabolism? Okay, well, you learned this in SCB 203, where amino acids are joined by peptide bonds. Okay, and this occurs in protein synthesis from DNA of a gene. Okay, as you learned from the nutrition lecture, the body can synthesize 11 amino acids by adding an amino group to a carbon skeleton. And of course, the remaining amino acids are essential and it must be supplied in the diet. However, unlike carbohydrates and lipids, proteins are not normally stored as an energy source. Okay, and so any excess protein is converted to glucose or fatty acids for storage. So let's look at the big picture of nutrient anabolism. Okay, so glucose through glycogenesis forms glycogen. Okay, however, glucose can be interconverted to lipogenesis to produce lipids. Okay, fatty acids through lipogenesis produce lipids. Okay, glycerol can be through lipogenesis form lipids, but it can also enter gluconeogenesis to form glucose. Amino acids, of course, can produce proteins, but it also can be converted into gluconeogenesis to form glucose. So to me, and this is pretty amazing, that there's an interconversion between all the major organic molecules within our human body to allow them to be, the metabolic rates to be interconverted. So this is the end of lecture 27.